This is episode 9 of the Immunology Podcast, Lymphocyte Activation with Dr. Marc Jenkins. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today, we have an absolute pillar of the immunology community here on the podcast. We are talking with Dr. Marc Jenkins from the University of Minnesota about his work in understanding lymphocyte activation in order to improve vaccines and prevent autoimmunity. We've also got our usual roundup of recent highlights in immunology news coming up, but first...
Are you interested in differentiating human pluripotent stem cells into monocytes? The STEMDiff monocyte kit from STEMCELL Technologies generates millions of monocytes, ready for downstream assays or further development into macrophages or dendritic cells. Learn more at stemcell.com/monocyte kit.
Well, hey there, Brenda.
Hi, Jason. How are you today?
Good. Are you ready for a T-cell day?
Yes, excited. Very exciting day.
Excellent. Well, I know our interview later is all about T-cells, but I think we have a few papers that cover this topic today too, don't we?
Yes, we do. I also have some non-T cells, so just to keep it balanced. But yeah, T-cell day today.
All right. Well, why don't we hit the T-cells first, and then we'll come back and do all those other important things. I guess I'll start. I have one about immunometabolism, another trendy field where you combine two important words, immunology and metabolism, and make a whole new field out of the both of them together, which is how you get grants. So
easy.
Yeah, that easy, right? Just immediately get it. Outstanding scores, 10s all around from NIH. There you go. One and done. No, but in all seriousness, this is a cool paper. It's called Metabolic Control of TFH, that's T follicular cells, and humoral immunity by phosphatidylethylamine, or PEA. So this is by Gutong Fu, is the first author. It's in Nature, here published July of 2021. And what's really cool about this paper is it shows that PE or PEA, it goes by both, but we'll use PE, shorter, phosphatidylethylamine. So PE is one of the lipid components of the membrane, but it's also a secondary messenger molecule, but it's kind of in the same realm of PIP2 and PIP3, but not the same, but kind of in the same general family. And they really linked they tried to really establish the link between the regulation of this pathway and T follicular cells. And so they actually did a CRISPR knockdown screen and found out that the pathway involved in the metabolism and production of PE is very important for the development of TFH or T follicular helper cells, and that you have to have it for humoral immunity to properly work. And what's interesting about PE is that it's usually on the inner leaflet of the membrane, but can be on the outer leaflet. And they established that it's the presence on the outer leaflet and its co-localization with CXCR5 is really driving this process. So they show that de novo synthesis of PE through a specific pathway, so the cytidine diphosphate CDP PE pathway, and there's several pathways that can affect PE, but they show that through this pathway, that this pathway prevents internalization and degradation of CXCR5. And then if you delete one gene, PCYT2, which is one of the synthesis genes, versus PCYT1A, and that second one is a different pathway. It's the CDP choline pathway. So only if you really hit the PE pathway controlled by CDP. What ends up happening is activated T cells do not differentiate into follicular cells, helper cells. And then you have reduced humoral immune responses. And it's done by post-transcriptional mechanisms. This is all based on the signals coming in, synthesizing the lipid, and that prevent and maintaining stabilization of CXCR5. So it's pretty cool. Anytime you work with lipids, which I've had to do with my prior work, it's a pain. They can be very hard to work with. Extracting and quantifying lipid levels from membranes of cells is always finicky. And so being able to have a good read of that is hard. And so they were able to do that here. They used a mass spec after lipid isolation. You can also use mass ELISAs. There's different ways to do it. And they were able to do that and really show that in various knockouts, there's different levels of lipids. They did staining and showed the lipid co-localization because they can use a fluorescent version of this lipid that's uptaken and then show the co-localization of it with CXCR5. And it was pretty neat. So they were able to really link a very specific lipid metabolism pathway with downstream effects for differentiation of T follicular helper cells.
Very interesting. And of course, CXCR5 is one of the major markers for follicular helper cells. So that makes a lot of sense. But how exactly do they dive deeper into how exactly PE stabilizes CXCR5 in the surface?
So it co-localizes with it. So CXCR5 is found in regions of high PE. And so they do some really crazy co-localization experiments using TIRF, so total internal reflection microscopy STORM. So this is one of these super resolution technologies to, break some optical limits and have better point-to-point co-localization analyses. And so they're able to take that microscopy and really show that the PE is co-localizing with the receptor. And so it's it being there that's preventing its internalization. So the lipid prevents the internalization of the receptor. How the cells know to internalize receptors in the absence of the lipid, they don't know. But already at that point, you're getting into a whole other set of pathways. But the lipid is preventing by co-localizing its internalization.
And do follicular helper cells have higher levels of PE in their membranes than other CD4 cells? Do they look at that too?
Ooh, I have to remember that one. I think they do. I think they are actually upregulated, and they show that. That's one of the later figures, one of the extended ones. So they show it's on the exterior surface more. So PE is in all cells, but they show that there's more of it on the exterior surface on T follicular helper cells. And accumulation of it occurs during T follicular helper cell differentiation, but not Th1 cell differentiation. For anyone who wants to see that, that is extended data figure 60
million.
Oh yeah, there's 16.
I mean, these papers that have just pages and pages of supplementary data, it's sometimes can be very daunting to look for the details among all those figures. I just wonder, I ask these questions because I wonder, are we having kind of a chicken and egg situation? Is a follicular helper cell kind of drive the accumulation of PE on the membrane to stabilize this very important receptor, or does the presence of PE I guess it would say is required to generate follicular helper cells?
Correct. If you knock out the PE pathway, you don't get follicular helper cells.
So you really need that in order for the development of this subtype.
Yep.
Okay. Very interesting.
And the CXCR5 signaling downstream. So if you don't have if you put CXCR5 back in or stabilize it, you can override this. But if you don't, then it won't be there, and then it doesn't work, and then you don't get follicular helper cells.
Well, why don't we move now from metabolism to microbiome and talk about a paper that was published in Cell, coming from first authors Crystal Nangao Makamdop, Arti Tala, and Ashish Sharma, shout out, from the University of Minnesota, Emory, and the NIH. And that is titled Translocated Microbiome Composition Determines Immunological Outcome in Treated HIV Infection. In this publication, the authors follow a cohort of 11 patients that are treated with antiretroviral drugs after being diagnosed with HIV infection. So as a little bit of background, so I think our listeners probably know that HIV has a particular tropism towards CD4 T cells, and infection with this virus results in a pretty much depletion of CD4 T cell populations in patients. And this has, of course, devastating consequences to the immune system and also to the integrity of the mucosal barrier in the gut, because all the cells that are there keeping homeostasis are not there anymore. So patients also show translocation of bacterial products across the gut barrier and general alterations of their microbiome, dysbiosis, and this contributes to the comorbidities that are observed in AIDS, and a general systemic inflammation, which is, of course, detrimental to the quality of life and the survival of patients with HIV. So combined antiretroviral therapy, or CART, is our current standard of treatment against HIV and can have really, really beneficial results. So patients have almost pretty much undetectable HIV levels upon treatment, and they can reconstitute and recover their CD4 T cells throughout several months of treatment. So this, as I mentioned, this paper, they follow 11 patients, and they're in Uganda, and they see that they can achieve virus suppression around month 4 for most of the patients. And when they look into this, they look at the cytokine and chemokines in the plasma of these patients, and it's very interesting because they see a fluctuation of different chemokine and cytokine groups that kind of tell a little bit of a story of the treatment. So what they see is an initial pro-inflammatory cluster that includes IL-1 beta, IL-6, IL-8, and that is increased following the initiation of the treatment. And then they see also around 1 year, they see a progressive increase of IL-17 and TNF-alpha, which then decline when they measure again at the 2-year mark. But by this time, there's a third cluster of cytokines, which is prevalent, which is a more homeostatic TH1, TH2 cytokine cluster, interferon gamma, IL-2, IL-4, IL-7. And they see that this is what they observe in patients that then recover their CD4 compartment around the 2-year mark. And the authors have a model which is also supported by other publications, in which this initial inflammation, which probably is driven by the presence of all of these microbial products that are translocating into the blood, and is really inducing innate immunity, and then a more Th17-like response around the year 1 favors the recomposition of the mucosal surface and a kind of more sturdy protection against this translocation around the year 1. And then around year 2, what they see is a normalization of the cytokines of the immune system and a reduction of this innate immune response, which of course would be detrimental in the long term. And in fact, what they see, and others too, is that the initial inflammation correlates with a better reconstitution of CD4 T cells, but if this inflammation persists into year 2, it has the opposite effect. And when they look into the microbial diversity, and for this they look into they do deep sequencing of DNA and RNA in the plasma, so they look at translocation of microbial products and microbial DNA and genetic information. And they see that of also this values on the composition of the microbiome also fluctuates throughout treatment. And they zero into, in this case, a particular genre of bacteria, which is the Serratia, which associates with an increased pro-inflammatory and Th17 cytokine levels, particularly at the beginning of treatment. And they also, what they see is that also in vitro, when they co-culture these bacteria or products from these bacteria with PBMCs, they also see that they can induce the production of IL-1 beta, IL-6, TNF, and this is kind of specific of this particular genre and not others, and it probably has to do with specific lipopolysaccharides that are found in this particular species. And so long story short, because they do a lot of analysis and they also look into patients from other countries, from Canada, for example, that have probably different microbiome compared to people from Uganda. And they conclude in a model in which CD4 T cell recovery is benefited by an initial pro-inflammatory cytokine milieu, a reconstitution of the gut barrier with a Th17 response, but then this is normalized around year 2, and only then can a proper reconstitution of the CD4 T cell compartment be possible. So, and I think it was very interesting and opens a lot of avenues in treatment and understanding whether maybe particular bacterial species could be beneficial for favoring recovery in patients treated for HIV infection.
So that's really interesting. I think it goes, obviously know my belief about how important the microbiome is. It's interesting though with chicken and the egg with it, right? Like, so is this, they're showing some level of causal effect and the bacteria is going, but the key experiment would be switching the microbiome in a person from one to the other and seeing if it could have an effect. Obviously that is probably, you can't really do that with people easily. And so that's kind of out of the way, but it's interesting to see that this is happening. I think immuno-oncology is now having a heyday with the microbiome, and it's interesting to see that this is coming to other parts of immunology as well. I mean, it's obvious that I think it's obvious. It's been obvious for a long time the microbiome tones the immune system, but how it's having effect on all these other processes is, an exciting time, frankly.
Yeah, for sure. And then I guess the question is whether you can use this knowledge to, I don't know, put some probiotics or something like that on patients to improve their recovery in the long term. I think we're still far from that, but this is definitely a step in the right direction.
I completely agree.
So what else are we going to talk about today?
Oh, well, I have one for you that is an interesting one. This gets really into some biophysics here and some chemistry. So it's interesting. This is also in Nature. Its title is "A Condensate Hardening Drug Blocks RSV Replication in Vivo," from Jennifer Riso Ballester. Nature, also July 7th, 2021 here. So RSV, respiratory syncytial virus, is a virus that really hits kids hard. It's a major pathogen. And what they found here that was really interesting is that it's the type of virus that likes to replicate in inclusion bodies. And they found actually that some of these viral inhibitors they're looking at, in this case, it's a peptide A3E, it's a steroid alkaloid cyclopamine. And there's another chemical analog related to it called AE3. So these small molecules actually affect the inclusion body, and they're known to work, but there's some side effects with some of these because they affect the hippo pathway, and the hedgehog pathways and a bunch of other stuff. Actually, make sure it is the hippo, but it's hedgehog. So they affect the hedgehog pathway, which is why they've had trouble being used in kids, because you don't want to be inhibiting important differentiation pathways in children, especially young kids, because RSV is in really young kiddos, like infants. But they were able to then use this analog, which has less of that side effect, and they were able to delve into the mechanism. And the mechanism is what's key here, because they're able to really establish this mechanism, do it in a mouse model, and then have the analog, which has less of the off-target effect of affecting the signaling of hedgehog, but does have the inhibitory effect. And how it works, and this is what's crazy, is that these inclusion bodies are this condensate. So they're this weird biomolecular entity, like this phase transition condensate. And what they found is that this molecule disorganizes and hardens the condensates. And so by hardening it, it changes the properties of it, and they can't function to replicate. And the size of the molecules that can enter that condensate changes. So originally these condensates are pretty tight, but they find that it can shift so that 40 micrometer and then are intermediate, and 70 micrometer can enter. And so as a result, they really demonstrate that by altering these condensates, you can affect the whole thing. So this small molecule goes in, affects these inclusion bodies by affecting this phase of matter that they're in, and then prevents viral replication. And that's the mechanism. So what's cool about this is it's previously had been, this had been considered relatively undruggable. Inclusion bodies are hard to drug. This opens the size up, so you can then drug them a second way by opening the so to speak, the pore size into the structures. And then secondly, it alters things by if you can alter the molecule, you're not trying to find something that fits the binding pocket of RSV that could also be binding hedgehog. You're instead altering the properties to affect the condensate state. And that's what matters. So it's basically opening up a whole new drug pathway through basically biophysical mechanisms.
Are other viruses that are also using the same condensate mechanism?
Yeah. So they think, so inclusion bodies is something that's commonly seen in negative strand RNAs, and they're generally shown to be biomolecular condensates that form through phase separation. And so they're thinking this can be a general principle that applies outside of just RSV.
Are they looking into human treatments, or what are the limitations?
So they started with
of that
mice,
study?
and I think what they want to do next is find a better drug that has less off-target effects in mice that they know about, and then go into people. That seems to be where they're going here. But they showed that it worked in the mice. And then the next step is to find a better drug. But now it's basically opened a new category and mechanism of action, which is cool. And those are
Right,
measurable, right? You can measure the condensate state. You can measure the pore size. So you can start screening compounds for that property, not inhibiting viral replication, for instance. Just work on the condensate and then go from there.
Very interesting. I guess that these very key aspects of the viral replication pathway are obviously very clear targets, but if they're happening so deep into the cell, must be in principle hard to target. You only can make very small molecules that can easily diffuse or be transported without problem.
Exactly. And what's interesting is that it as far as I know, and maybe I'm wrong and someone will at me, but humans don't use condensates for a lot of their, RNA or DNA or
Right.
protein replication, right? That's not a structure. And so you can target something like that. And that gives you that, that nice thing we like in infectious disease where it affects the pathogen, but not the host. That's why you can get lots of antibiotics that don't have off-target effects as much because, we don't have cell wall synthesis in the same way. But
Yeah.
the caveat being that that's been hard with some of these viruses and inclusion bodies to get them in, and you have off-target effects, and now they're finding a new mechanism of action, which is why it got in the nature. And it's pretty cool. They do some really cool experiments in here to get to it. A lot of fluorescence, some biophysical property measurements of these things, hypotonic lysis, all of that good stuff.
Very interesting. I hope that they can continue into a human treatment because VSV can be a very devastating infection for young children and for, of course, the immune compromised.
So yeah, so RSV really affects sick kids. It's one of the things I saw a lot of people get hospitalized or children get hospitalized, babies with RSV. And so, but then they're really sensitive to any of the off-target effects. So you have to have an even better tox profile because it's a kiddo. And that's been hard with RSV.
So to finish up today's roundup session, I want to talk about the latest installment in COVID research for the vaccine-induced immune thrombotic thrombocytopenia, a saga, which has been really a headache for AstraZeneca and Johnson & Johnson and their adenoviral vector vaccines. So this story comes from the journal Nature. It's still under edit. It's been published ahead of print. And it's titled Antibody Epitopes in Vaccine-Induced Immune Thrombotic Thrombocytopenia. First author Angela Huynh from McMaster University in Ontario, Canada. So vaccine-induced immune thrombotic thrombocytopenia, or VIT, as I will refer to it from now on, is a very rare adverse effect of the adenoviral vector vaccines and results in, as the name suggests, thrombosis, so blood clots, particularly this rare cerebral venous thrombosis, also severe thrombocytopenia or kind of depletion of the platelets in the blood, and which usually develops between 5 to 24 days following the first dose of the vaccine. And this has been observed only for the AstraZeneca and the Johnson & Johnson adenoviral vector vaccines. At the beginning, the first patients were reported around April, and it was really, I remember, a lot of confusion around this syndrome and a lot of preoccupation, a lot of concern And very sometime after the first cases were reported, there was another syndrome that was identified as being very similar to VIT, which is known as HIT, which is heparin-induced thrombocytopenia. And in this syndrome, which has been more characterized and has been known for many years, patients that are treated with heparin develop antibodies against platelet factor 4, which is a small cationic protein that is contained within platelets, so it really goes around. And the exposure to the heparin somehow, so the heparin somehow, probably due to their structure as a large anionic polysaccharide, exposes certain epitopes that are usually not available and induces a humoral response or exacerbates a pre-existing humoral response against these antigens, these epitopes that are hidden in this PF4 proteins, and ends up clustering, and these antibodies cluster PF4 molecules and generate big clusters that end up binding and cross-linking Fc receptors on the platelets themselves, and this causes activation and coagulation because this happens throughout the whole body. This is a very systemic response. So one of the first things that people saw is that anti-PF4 antibodies are in fact found in patients with VIT, and this made this kind of connection very clear. So what they make in this paper, in this study, they take 5 patients with VIT that after receiving one single dose of AstraZeneca, and they look into the properties of their antibodies and trying to understand where in PF4 they're binding and how do they compare to a HIT antibodies. And of course, they see that in fact, there are antibodies recognizing PF4 at high levels in these patients. And after doing a lot of kind of structural analysis, and they also do kind of what they name as alanine scanning mutagenesis. So they are doing all these very diverse mutants of PF4 in which they replace different amino acids with alanine, and trying to understand which are the crucial amino acids for the binding of these antibodies. And so what they see is that the antibodies from VIT patients bind to don't bind to the same places as HIT antibodies. They have usually a higher binding response, and they also bind to the areas in which are usually bound not by antibodies but by heparin in the case of HIT patients. So basically what they do is they characterize the antibodies. What is not clear yet, I think, is what exactly induces the upregulation or the production at such high levels of these antibodies against PF4. I think that is still not clear. Might be kind of some kind of immune activation from the adenoviral vector, from the DNA that is inside the adenovirus. But regardless of that, they show that these antibodies can bind to PF4 in both places that already had been described for HIT, for heparin-induced thrombocytopenia, but also other sites that are specific for this syndrome. And they suggest that then one of the potential treatments are high level of high dose immunoglobulin in an intravenous injection that can compete with the IgGs for the Fc receptor, and then reduce the cross-linking of it on platelets, and then reduce platelet activation. And they also see that heparin, in this case, would compete for binding with these antibodies as well. So I think in general, I think it's a really good final piece of understanding the mechanism of action of these antibodies. And I hope that this will lead to some treatment options for patients that unfortunately find themselves in this situation.
So that's good to know that they're able to start linking these and get to it. I wonder, my only real question on this one, were they able to show that in the case of VIT, it's mostly with the adenoviral vectors. Were they able to show that this epitope doesn't appear in, say, the mRNA vaccinated individuals? Did they do that comparison and show that the epitope is only in adenoviral individuals?
Not themselves, so they don't do it. They don't have any patients with mRNA vaccination. But epidemiologically speaking, there are no events of VITs with patients that have been vaccinated with the mRNA vaccines. And there is a clear, statistically significant increase in these events on people vaccinated with both AstraZeneca and Johnson & Johnson, which really points to the adenoviral vector having something to do. So one of the theories is that the double-stranded DNA can be released and can be part of, in the same way heparin binds and generates these complexes, the same can be, for example, the DNA that is contained within these adenoviral vectors.
Got it. All right. Well, I mean, I'm very happy that we have a COVID paper again. We skipped one, so we got to get back into it.
Yes.
Excellent. Well, off to T-cell land. Here we go. We're going to be speaking to Dr. Marc Jenkins at the University of Minnesota in just a moment. But before we get to that, decorate your lab with a Nature Protocols wall chart outlining the production of CAR T cells for therapy, from apheresis collection and T-cell enrichment to gene modification, expansion, and delivery. Request a free copy of the wall chart at the STEMCELL Technologies T-Cell Therapy Resource Center by visiting www.stemcell.com/T-cell-therapy.
We are joined today by Dr. Marc Jenkins, who is a professor at the Department of Microbiology and Immunology and director of the Center for Immunology at the University of Minnesota. The focus of the Jenkins Lab is on CD4 T cell and B cell activation, and these are fields to which Dr. Jenkins has done really huge contributions. Thank you so much for joining us today at the Immunology Podcast.
Yeah, great to be here. Thanks.
I was hoping to start our conversation today with some of your early research as an immunologist who started in the 2010s. I take, for example, CD28 activation for granted. Now that I realize it always existed, we always knew what CD28 did. But I think a lot of our listeners will be very interested in listening or hearing your experience actually being the first person to find out and publish what CD28 does and how important it is as a co-stimulatory molecule for T-cell activation. I was hoping you can maybe take us with you to that time and what was the state of the art of T-cell activation back then in 1992, and what have we learned since then?
Yeah, great question. Well, that I worked on co-stimulation or on T-cell activation when I was a postdoc at the NIH with Ron Schwartz. And at that time, the thinking in the field was that, and this was, not too long after the T-cell receptor had been discovered. I started doing this work in 1986. And so, the discovery of the T-cell receptor by Mark Davis and Steve Hedrick was, just incredibly exciting to actually understand, how T-cells recognize antigens in the form of peptides bound to MHC. There were also discoveries around that time from Howard Gray and Emil Unanue and others that actually demonstrated that peptides bind MHC and that there's a single ligand for the T-cell receptor. And so this was, just took T-cell biology from the dark ages into the light. But there was then an incredible focus on the TCR. And so a question that I was addressing in my research as a postdoc was although we knew the TCR was necessary to activate T cells, was it sufficient? Was that the only signal you need to get a T cell to become activated? And so in my research, I was working with a chemical fixative that basically fixed antigen presenting cells, killed them, turned them into little billiard balls, but their surface molecules were intact. And what I found was that they could present antigen to these T cell clones that we were working with. We could show that the T cells got larger, but they didn't proliferate, didn't make cytokines. And if we came back then and tried to stimulate them with viable antigen-presenting cells and antigen, they also didn't respond to that. So that led to two concepts. One, the TCR, although it's necessary for T cell activation, is not sufficient. Antigen-presenting cells do something else. And if they don't do that something else, the T cells actually become unresponsive. So this idea of T cell anergy. So, and then, of course, that spurred a tremendous interest in what the something else was. What was the other signal that antigen-presenting cells provide to T cells other than present peptide MHC to TCR? And I was highly influenced when I was still in Ron's lab, but then as I started my own lab in a study that Carl June had done, I saw he was one of your other guests, and Carl had found that stimulating human T cells with this chemical called PMA, a 4-ball ester, could give this kind of weak T cell activation that wasn't blocked by cyclosporin A, which we knew at that time was a TCR blocker. So this, and we had found that we could take antigen-presenting cells and PMA and stimulate weak T cell proliferation. We could show that that wasn't blocked by cyclosporin A, and Carl could show that with an antibody against CD28, or what was called TP44 at that time. And so I, figured that TP44, or what we now know as CD28, was a great candidate for the receptor on the T cell that could be receiving this other signal. And so we did some experiments with human T cells that were specific for tetanus toxoid, and could show that CD28 signaling produced a very powerful synergy with TCR, allowing full activation of the T cell. And Jim Allison at that same time, using a mouse model, showed that that CD28 signal prevented anergy. And so this work, really, led to this idea of the two signal model that had been proposed by Kevin Lafferty before, and by Mel Cohn and Peter Brecher. But this, was actual experimental evidence for this kind of idea.
So what led to the initial thought experiment, or, let's take a step back. Obviously, when you look at something in hindsight, it seems like a linear story where there was an idea and then you solved something and it was big and great and discovered, and then it goes on, and all of us in the bench know that's often not what happens. What led for you back then to the original idea? Is a TCR, as you said, necessary, but is it sufficient? What led to the question of, is it sufficient? And you mentioned Carl June's work got you down the path in part because he found something else that was having this effect. But what led to that question of, is it sufficient, that drove this?
Yeah, at that time, the easiest way to study antigen-specific T cells in the mouse was through what are called T cell hybridomas. They're the T cell version of the kind of hybridomas, B cell hybridomas that make monoclonal antibodies. You take a T cell that has a specific TCR, you fuse it to a tumor cell, it proliferates forever. But if you had the right antigen with antigen presenting cells with the right MHC molecule, you could get those T cells to make IL-2. And so it looked like from these hybridoma cells that was really the only signal they needed, was just this signal through the TCR. And then we tried to apply that idea to our T cell clones that weren't transformed. They're regular T cells. They wouldn't proliferate unless you stimulated them, and found that, through various kinds of experiments, that when we just gave that pure TCR signal, they didn't seem to proliferate. So there was a disconnect between what these tumor cells would do and what the normal T cells would do. And so we were trying to figure out, well, what is that? What is that difference? What is that thing? And there was also a large literature at that time about interleukin-1, which was one of the few known cytokines at that time, that it might have this kind of co-stimulatory activity, that it was TCR plus IL-1. And so that idea was out there bouncing around as well. but we could quickly show that, in our system, IL-1 didn't do anything. And so, and we had other complicated cellular immunology experiments where we could provide this co-stimulatory signal on a different cell than what was presenting peptide MHC. So we could work out some of its properties. So we knew that the ligand was on the surface of the APC, it wasn't secreted. And of course, that turned out to be CD86, the ligand for CD28. So that's kind of the way because Ron Schwartz's, idea was to use artificial membranes and then just put in TCR and then everything else that's needed and produce this kind of artificial antigen presentation system. But to do that, you need to know all the things to put in. And other people like Mike Dustin and, did that well after Ron's in our work. But so it was, this drive to figure out the minimal activation requirements.
Can I ask, at what point was TCR stimulation with peptide, I don't want to say replaced, but complemented by anti-CD3 antibodies for stimulation of T cells?
Mm-hmm. Yeah, I think that came from expedience. To do the kind of experiments that we did in those days, you had to have these T cell clones that you could grow in vitro, maintain them without dying, becoming senescent. There weren't it was in the early days where it was hard to get antigen-specific T cell responses just from like primed lymph nodes in mice or from PBMC. And so once it was clear that, in the old parlance, TCR was signal 1 and CD28 was signal 2, and there were antibodies against the agonistic antibodies against these targets, you could just do a lot of experiments with, just any old T cells from animals or humans. And so that did really drive the field because it was so easy for anybody to do.
And I think this kind of takes me to this next thing I wanted to discuss with you, which is after this publication, and I think in general in your work, you have focused a lot on CD4 T cell activation and the role of CD4s and in the activation of B cells. And I wanted to ask you, what do you think are the main challenges when studying CD4 T cells and their differentiation, their activation? Because in the CD8 field, sometimes I feel like the readily available, very efficient multimers or tetramers that are used for picking up the cells, and the kind of less complex differentiation of the cells make for a slightly different field of study. What has been your experience by understanding the cells and their specificity throughout your research?
Yeah, you hit it right on the head. Everything has been harder to study CD4 T cells. One of the things that the main things is that we've done studies now, and other people have done studies which basically show that a single naive T cell will make about 1,000 effector cells, let's say for argument, when you stimulate it. A single naive CD8 T cell can make 10,000, so 10 times more. So any system or in any experiment where you're trying to identify antigen-specific cells, it's going to be 10 times harder for CD4 cells because they're 10 times more rare. And so these limit of detection problems for CD4 T cells are a huge, huge issue. And then of course, the state-of-the-art way to find antigen-specific T cells is with peptide MHC, tetramers, or multimers. So for class 1 molecules, you can make the class 1 heavy chain and beta-2 microglobulin in bacteria. You can make huge quantities of them, purify them to homogeneity, mix them together with your peptide of interest, they reform. You can get these beautiful monomers that you can then biotinylate and make tetramers, and in huge quantities. So for most class 2 molecules, You can't express them in bacteria. They don't form properly without proper glycosylation. So then you have to use mammalian or eukaryotic expression system. The yields are lower. It's harder to figure out what the minimal peptides are because, class 1 basically binds a 9 amino acid peptide. For CD4 cells, even though the core that binds MHCII is 9 amino acids, there's all this stuff hanging off the ends. So it's harder to figure out what the real epitope is. The peptides, because the groove of class 2 is open, the peptide can, within, let's say, even like a 20 amino acid peptide, can have 3 different 9 amino acid cores that bind in 3 different registers. If that happens, you basically have 3 totally distinct epitopes within that. And when you try to make a tetramer, you have streptavidin that's got 3 different kinds of arms. Very few molecules have 4 identical arms. One nightmare after another, technically. And so for a while, T-cell receptor transgenic mice leveled the playing field because you could make a mouse that had a monoclonal repertoire of T cells that were CD4 T cells and saw a known peptide MHCII ligand. And so we developed this method of taking a few T cells from a mouse like that, putting them in a normal mouse, and then tracking them after you give the antigen. And so you could elevate the frequency a little bit by that adaptive transfer trick, solving the rare, cell problem, and then they would expand enough you could find them, and that was really, a boon to the field. You still see that done all the time by people, but we, inspired by the late Leo Lefrancois' discovery, that although it's incredibly useful when you artificially elevate the number of T cells, you paradoxically get some abnormal T cell activation, mainly inefficient activation, because all these T cells compete with each other for a fixed amount of peptide MHC. And so that really drove me, even though I loved, adaptive transfer of T-cell transgenic cells, that was pioneered in my laboratory. We try not to do that anymore. And so study the real repertoire with tetramers now. And then the final insult is that when a CD8 T cells recognizes peptide MHC1 with its TCR, the CD8 molecule binds to the alpha 3 domain of that class 1 molecule. It kind of helps the binding. So when you do peptide MHC1 tetramer staining, you get this affinity boost from CD8. Well, the affinity of CD4 for the analogous site on class 2 is at least 200 times weaker. And so peptide MHC2 tetramer binding to CD4 T cells does not get aided by CD4. And so tetramer staining isn't as good. So, at every level, it's been harder. And that has really caused the CD8 T cell, the study of CD8 T cells to dominate most of our understanding of fundamental concepts in T cell biology, related to effector cell expansion, immunological memory, and things like that. CD4 cells lag behind. And then, of course, they also have this confusing, really cool, but as you point out, CD8s basically have one fate. They get activated, they differentiate, they make gamma interferon, they kill. That's a little too simple. But for CD4 cells, they can become TH1, TH2, TH17, TH9, IT regs, endless, not endless, but much heterogeneity, each with their own differentiation rules. And some, the T follicular helper cell, for which we still do not have an in vitro system to generate. So lots and lots of challenges.
So to go on with this a little bit, you had mentioned the tetramers that work really well for MHC1, but not MHC2. And I know one of the papers you'd put out recently was an engineered tetramer. And it was an interesting approach using a combination of CRISPR-directed evolution in these tetramers. So kind of 2 questions for you. One, could you walk through for the listeners at a high level what you guys ended up doing in that paper? And then secondly, did you guys gain any understanding about fundamental biology, seeing in the end what these modified tetramers were. Because you mentioned that the MHCII system, the T helper system, doesn't have the arm that binds as well. That's 200-fold less. And now you've made some tetramers that work better. Did that in that process? So how'd you make it? But did that process teach you anything about, structure function relationship? Because I always put my biochemistry hat on. That's my background in relation to, the T cell receptor system and MH2 that can be helpful going forward.
Yeah, great. That's you're right on, man. So as I mentioned, class 1 peptide MHC complexes can bind, when you make a tetramer, those can bind TCR. TCRs bind the peptide MHC groove, and then the CD8 molecule binds the stalk of the class 1 molecule, and that helps the tetramer bind. For CD4 cells, they have a TCR, they bind peptide MHC2, but their CD4 molecule binds the stalk of class 2 so weakly it doesn't aid tetramer binding. So we used molecular evolution, which is this incredibly powerful technique to screen a library of class 2 molecules where we had directed mutations to this site on class 2 where CD4 is thought to bind, and then using an iterative cell sorting technique with a tetrameric form of CD4, we found class 2 mutants that bind CD4 at about the affinity that CD8 binds class 1. So now when you make a tetramer from those class 2 molecules, you detect more cells in a polyclonal repertoire than you do with the wild type conventional tetramer with the same peptide in it. So it varies, but it's about twofold up to about fourfold more cells. So number one, we were missing, half the cells that were responding to the antigen, number one. And now we're doing experiments to say, well, was there anything special about the half we were missing? To get to your point, Jason, and we're doing that now. Those kind of experiments now. We have some interesting findings with regulatory T cells actually along those lines, but that's work in progress. So, even if we find these, and the cells we were missing probably had lower affinity TCRs, so they needed that CD4 boost to bind the tetramer. And so it's not crazy that those lower affinity cells could have different, get activated in a different way because they receive a weaker TCR signal on average. And so we're trying to sort through that all now. But if nothing else, we've made it twice as easy to find the relevant cells. And so, as I pointed out, that alone, I think, is going to help. And the other thing is that because these peptide MHC1 tetramers bind better, they've actually are able to bind to tissue sections. So you can use them to do anatomic studies to see, these CD8 cells that see this tumor antigen are either in the tumor, they're on the edge of the tumor. Class 2 tetramers, because they bind less well, historically can't bind tissue sections. So we have some preliminary data now that these evolved tetramers can do that. So that's going to open up some nice ways to study the anatomy of the CD4 T cell response that heretofore we could only do with artificial TCR transgenic adoptive transfer experiments. So
I have so many questions, but I would like to first ask, how did you solve the other issues with making MHC2 tetramers, particularly regarding expressing the proteins and loading those peptides?
Yeah. Well, John Kapler and Jerry Nepom and some other pioneers had really figured out ways to express MHC2 molecules in eukaryotic cells, usually with viral expression systems, and baculovirus, I think, is what the Kapler lab normally uses. And so we piggybacked on their technology to get that going in our lab. We made a few tweaks, but most of the credit would go to them for that aspect of it. The thing that we really did add, though, was this idea of using tetramers to enrich the rare cells. in the same way you can use magnetic beads to isolate rare populations of, some very rare cell of any kind, like a stem cell. We could use that with tetramers then and enrich the antigen-specific cells from huge numbers of cells, from like all the cells in the mouse, and then enrich them so that we could detect basically every tetramer binding cell by flow cytometry without having to pass, 300 million cells through the flow cytometer, total cells. So we improve the sensitivity by about 100-fold with this enrichment technique, using tetramers that we make ourselves, but based on their technology.
And I don't do much work with tetramers, so my understanding is that so you express these proteins in a eukaryotic system. Yep. You make them; they can be used as markers. But then, do you also have to load a then antigenic peptide into them as well? And do you guys is the tricky part the tetramer and not the peptide, presumably? So you can then swap out different peptides later on and see if different peptides with your super tetramer do different things. Is that kind of the next level?
Yeah. Well, again, all those things have been problematic. In an actual antigen presenting cell, the class 2 molecule gets made with the invariant chain peptide bound in the groove. And then there's this other class 2 like molecule, HLA-DM or H2DM, that basically removes CLIP and allows a new peptide, let's say from a microbe or something, to bind. So, if you express like we do with these MHCII molecules in insect cells, they don't express HLA-DM. And so getting peptide exchange to work the way you can do it so easily with class 1 has been a really problem. You can do it for a few allelic forms of class 2, but for most, our favorite molecule, the class 2 molecule of B6 mice, where most of the gene targeted, strains exist, peptide exchange cannot be done without H2DM in the system. So we do have an exchange system now, but the way we normally do it is we use the trick of John Kapler to attach a peptide covalently to the beta chain. And then that will flop into the groove when the alpha chain of class 2 pairs with the beta chain. And then that peptide can't come out. But a peptide exchange system that works well for mouse MHC and for other allelic forms of human MHC would be a really important step forward, I think, in tetramerology.
See, there's a lot to be done yet, but it's very exciting to see that these new, these kind of tools are also starting to be more available for CD4 research, and hopefully will help us understand more about what determines the fate of a CD4 T cell. And in that topic, I was hoping to also hear from you. What do we know about how the specificity of the TCR, the strength of the TCR signaling, and in case of one of your very recent papers in which you look into particularly Th1 differentiation and this two-step model.
Yeah.
What do we understand about the different influences on CD4 T cell differentiation?
Yeah. Well, I think that the major interests, the major influence are cytokines from the innate immune system. Kind of as we've thought all along. I think it would be the innate cytokines that are present at the time that a naive T cell starts proliferating and then starts the differentiation process. And I think that but very early on, in almost every immune response we look at, the early progeny of naive T cells will start going down one of two paths. They will go down the T follicular helper cell path, or they will become a non-TFH. And then in the early progeny, as far as we can tell, split about 50-50. And then the half of that clonal burst that do not become TFH then are influenced by innate cytokines. Like if there's a lot of IL-12 around, they will become a Th1. And if there's a lot of, IL-33, they'll become a Th2. If there's a lot of, IL-6 and TGF-beta, they'll become a Th17. But that early bifurcation, the thing that, from our work, Shane Crotty, Troy Ranoff, a lot of people looking at this, suggests that the IL-2 receptor signaling through STAT5 is the key to that early bifurcation. And one of the main thing that controls the IL-2 receptor is the TCR. So you could imagine that naive T cells that have high affinity TCRs for the peptide of interest, that they would get a strong induction of IL-2 receptor, they'd get a lot of STAT5, and they'd be more likely to become a non-TFH, because strong STAT5 signaling is poisonous to TFH differentiation. So it cuts off that fate. So even in the context of innate immune cytokines, that early bifurcation might be driven to some degree by TCR signal strength, which could be inherent to the T cell because it's got a high affinity TCR, or it could be luck because that naive cell happens to be near an APC that has a huge amount of peptide MHC on it for anatomic reasons. And so but once you get a non-TFH, then it looks like it's really innate cytokines that are going to determine the fate of that cell. And in our recent work, a surprise that we got was that in a system that gives you, TFH and then Th1s, because the innate immune system drives type 1 immunity, there's an early step in Th1 differentiation in vivo that is IL-12 independent, and then gets reinforced by a second step that allows the massive outgrowths of Th1s, massively outgrowing the TFHs that form at the same time, and producing this kind of super Th1 that makes a ton of gamma interferon, and is actually important for protection against the kinds of microbes that Th1 cells protect us against.
On that note, you've also dabbed into Treg development and Treg differentiation. And talking about CD25 upregulation and TCR, the influence of TCR stimulation on IL-2 receptor, there's this recent publication, I think it was from the lab of Alexander Rudensky, in which they actually looked into that, how important it is, the strength of the TCR into upregulating IL-2 receptor, and that really drives the survival of Tregs during the thymic selection. It's really exciting. I don't know if you have any comments on that. I just wanted to mention it because it's really interesting how all this. You find these topics are so universal when it comes to CD4 CD4 biology.
Yeah, that's we don't do too much work in the thymus, but it's very clear that IL-2 receptor signaling is critical for Treg development in the thymus. And most of the peripheral Tregs that are Treg cells that are in the secondary lymphoid organs that have left the thymus, express CD25 and actually require it to stay alive. And so what's interesting is, because we're always trying to track antigen-specific cells. When you look at a repertoire of antigen-specific CD4 cells in the pre-immune repertoire, and for most of these, that is 1 in 100,000 T cells. But a mouse might have 300 of these cells. 30 of them are Tregs, and 270 are conventional cells. And those Tregs, they see a foreign antigen. Presumably, they get selected on some self-antigen with some high affinity or continuous signaling property. And they have CD25 like other Tregs, but when you give the antigen that population is specific for, those Tregs actually downregulate CD25. They actually respond to the antigen and start proliferating. And then as the antigen clears, they put CD25 back up and use it. So it's kind of the mirror image of a naive cell, which has, a conventional cell, has no CD25, gets activated, turns it on, uses it to proliferate. And once the antigen goes away, the CD25 goes away. So we're the we don't do a lot with Tregs, but we're interested in, number one, what do Tregs do in responses to foreign antigens? Why does every foreign epitope have an embedded Treg repertoire? And why is it always 10 times smaller than the conventional cells that see that same peptide? And then what do they do? Why have Tregs that are specific for a bacterial peptide? Isn't that counterproductive? And so we're doing experiments around trying to make repertoires that are missing only the Treg component of a repertoire specific for a single peptide, to then ask, how does that repertoire? But that's not easy to do. But we want to get at those kind of issues. So Tregs probably have a role in immune responses to foreign things like infections, and that role may be different from their role in preventing autoimmunity. Almost has to be. So those are some thoughts about Tregs.
So I had kind of a final philosophical T cell question for you. And this has kind of come up as part of this podcast. We review, recent literature in addition to talking with awesome scientists. And we noticed that everyone talks about, well, there's this new subtype of T cell or what have you, or, it has mixed function of Th1 and Th2 and so on and so forth. And so the philosophical question is, I know we know a lot of the work, and you were there, obviously, strong cytokine mixes really can push something to Th1 or Th2 or 17 or a Treg or a CD8 T cell. But I'm wondering, as we've gone on and how messy biology ends up being and likes to defy our ability to categorize things, how much of you think that in reality it's a spectrum, that the T cell that you're looking at is 80% Th1 and 20% Th2 in reality? And when us humans are looking at it, we're really just selecting the ones that are mostly one way or the other and calling it as one or the other because we categorize things, but that biology is not nearly so clean. So wondering, since you've been in this for so long, if you had thoughts about what's really happening, especially as all the single cell RNA-seq data comes out and everything else that we're seeing.
Yeah, that's a really good point. I think they, like any model, the early Th1, Th2 model, came from sledgehammer experiments. They showed extremes, extreme phenotypes. But I would say their utility was unbelievably high. The field really moved forward in a productive way. And so, but as the field has grown, as you mentioned, Jason, that we now with single cell RNA-seq, with now more other, flow cytometry basically is a single cell assay. So you can look for, are there any cells that express TBET, the Th1 lineage defining transcription factor, and ROR gamma T, the Th17 lineage defining transcription factor. And yeah, there are some cells that express both at the same time. You can prove it. And so how did that, why didn't the one lineage, defining factor suppress the other one? And so I think it's fair that that there are these mixed phenotypes. Like if you look at a type 1 immune response where you get Th1s, like in a viral infection, and TFHs, some of those TFHs express small amounts of TBET and B cell 6 at the same time. So Tregs, you have Tregs that have FOXP3, and in some infections or situations they have some TBET. Sometimes they have RO gamma T. So these mixed phenotypes exist. We're probably in a phase where the field is now going to chase all these gazillions of flavors. But I'm still impressed when the rubber meets the road, how robust the original kind of phenotypes are. And here's my evidence. The field of primary immunodeficiency and the field of monoclonal antibody-based therapy shows us that if you're born without an IL-12 receptor, you don't have Th1 cells, and you are going to get an opportunistic infection from an intracellular pathogen of macrophages. if you're on IL-17 antibody for your psoriasis, the main effect in your immune system, they're not going to have ROR gamma T positive cells, and your symptoms are going to get, that skin inflammation is going to get better. So the original paradigm still helps us understand immunopathology in humans. And so I get it. It's really important to understand these cells that seem to be going down. There was a fork in the road and they went down the middle. But I still think we can. The evidence to me of how useful a lot of the original polar phenotypes are is how helpful they are when it comes to disease, understanding extremes of immunity or immunodeficiency.
Really fascinating. And I think also the field is still evolving from those initial kind of breakthrough models. And as I've also been working through my PhD with the CD4 cells, Tregs, Th17 cells, and they're really amazing cells. I don't know how else to put it. And thank you really so much for going through this with us and talking about your experience in this field. I think our listeners are, I'm sure, extremely grateful for all the information. Here at the Immunology Podcast, we also like to finish up our conversations with a little bit of a question unrelated to research often. And so I wanted to ask you, when you're not conducting research, when you're out of the lab, what are you doing?
I'm either cycling. I'm an avid cyclist. I ride 11 miles to work and 11 miles home every day. And I don't drive very often anymore. I bike so much, and then we run onto one car, which is my wife's car. So I cycle that much. I'm addicted to it. Basically all forms of it. Although I've had a few pretty bad bike accidents, and I don't want to have any more. So I don't think I'll be doing much mountain biking. But I like cycling. I like touring. I like going as fast as my old body will go. And so that's a main avocation of mine. I also like fishing. I'm a Minnesotan. We have lots of lakes. So I like to spend time on those lakes trying to catch fish. I usually don't, but it's still fun to be out in the water. Or out on the ice. I fish in the winter, which I enjoy even more than fishing in the summer, believe it or not. So those are some of my avocations.
So related questions. I also fish. I grew up on the West Coast, but I'm now in Pennsylvania, so I'm getting used to bass when I used to do salmon. So first follow-up is, what do you fish? But relatedly, how does biking in Minnesota in the winter work? And then also, have you ever been able to bike to go fishing?
Oh, yeah. So the last question first. I've toured with my buddies where we do bike camping. And I have a collapsible fishing pole. So usually we're at a campsite on a lake. And so there's a fishing pier and I fish. So I do that a lot. And usually in Minnesota, the really prime fish to catch is the walleye, which is a very delicious freshwater fish. And I also fish for bass. We have bass, large and smallmouth bass. And then we have a large pike species in Minnesota called the muskellunge, the muskie. I fish for that. I don't know. I maybe catch one of those a year, but that fish can be 50 inches long, and that's a big, looks like an alligator. Yeah. So
I've done steelhead, which get up to 7 feet, and it's like pulling a dinosaur out of the water.
Yeah. So those are some
wow.
to do. And in biking and in Minneapolis is, sometimes Portland beats us, but we're often the number one bike city in the country. We have so many bike trails, and they keep them pretty much open in the winter. So for my winter bikes, I use studded tires, and that to grab the ice, or it's pretty nasty.
I didn't know they made that.
If I may say, you're I assume you're aware that Amsterdam is biking capital of the world. Maybe not all terrain. Everything's pretty flat here, but for nice views from next to the canals, you can always come here with your bike.
Yeah,
in good company.
I would love to do that. Although I've been to Amsterdam many times, and I'm just amazed at how many bikes there are near cars, and how it just it would be scary for me to be cycling in Amsterdam. And maybe get out away from town and out touring.
No, but the cars just are behind the bike. So the bike's first and the car's second. That's how it works here. So no worries.
That's good. I'm just afraid if you tried that in Boston, you'd
oh yeah,
get over.
Combat driving. Let me tell you. Oh Lord.
Yeah.
All right. Well, thank you for coming on today. It's been an absolute pleasure.
Yeah, absolutely.
Enjoyed it. Thanks for listening to my opinions.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at immunologypodcast.com to get show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on Twitter at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.